I've travelled here to Valencia in Spain to meet arguably one of the most important and controversial figures of science in our generation. I am talking about J. Craig Venter. From initial studies looking at the way that neurotransmitters work in the brain, he moved on to an interest in genetics and then genomics. It was a career path that was to lead him in the year 2000 to the White House, where he shared the praise for one of the greatest undertakings in human history, the sequencing of the human genome, unlocking our own genetic blueprint. Despite an undistinguished background as a student, he has become one of the most famous geneticists of the early 21st century. Dr. Venter, pleasure to meet you.
Nice to meet you.
Craig Venter, it's a pleasure to meet you. Your autobiography, A Life Decoded, and one of the stories in it which intrigued me was how you got into medicine and then into genetic research, and that came out of your experiences in Vietnam.
Well, I did very poorly in school and left school at age 17 to take up a surfing career, but interrupted very abruptly by being drafted into the military, because this was at the start of the Vietnam War.
Yeah.
And I ended up being a medic in Vietnam, and it transformed my life. You know, I saw the value of education, I saw the value of knowledge. The more knowledge I had, the more lives I could save. Well, being faced with things like that almost on a daily basis forces you to either find a way to think about it or ignore it, and I chose to think about it and try and understand it, and that led to wanting to go into medicine, then actually changing, being really excited about science.
One of the things that comes out very strongly in your autobiography is that you are an innovator in the field of genomics. And I think perhaps the most influential technology that you developed was shotgun sequencing of DNA. How does that differ to what they were doing before?
Well, everybody thought genomes were too large to be tackled as a whole. So it was envisioned to break them into thousands or tens of thousands of smaller projects and distribute them around a country, around the world. For example, the yeast genome was one of the first projects funded by governments. It took over 1,000 scientists over 10 years to do that genome. And we reduced that 10 years and 1,000 scientists to 4 months and a handful of people.
And I mean, I think the original budget for the Human Genome Project, which you were heavily involved in, the old way was $3 billion.
That was just the U.S. budget.
Right.
The U.K. and the Wellcome Trust probably spent at least another $1 billion. We did it in 9 months for $100 million, which is still a lot of money, but right. Compared to $5 billion, that's a pittance.
And you did that via sort of the private route, and I found that very, very interesting. Is that a reflection of the fact that you went to venture capitalists effectively, a result of any disillusionment about the way science was funded?
It was a reflection that we couldn't get the money from the government. They had their bureaucracy set up, and it was only because it wasn't venture capitalists, it was a company that made the sequencing machines that put up all the money, more as a test to show their sequencing machines would work.
Is this applied biosystems?
That's right.
And I noticed also in your autobiography, one of the things that you said is that when you set up your various institutes, Tiger and Celera and all the rest of it, the two things that you insisted upon was no tenure, and you wouldn't allocate funding, you wouldn't allocate space on the basis of funding.
Yeah.
And this is still very common in the United Kingdom, certainly in the tertiary education sector.
Yeah.
What made you go down that route?
Well, it's still very, it's the standard in most American universities as well. It's, you know, tenure used to be something that protected risk takers. Now it seems to be something that protects mediocrity. And so the whole purpose of it has gone away. And in science, your tenure should be the quality of the work you do. And so even Nobel laureates like Ham Smith don't have tenure. We're all at-will employees. I don't have a contract even with my own organization because it's the quality, when people do world-class research, that's better job security than any contract you could have with somebody.
Just tell us a little bit, if you would, about how it felt to be in the White House in 2000, because there was a lot, it seems to me, of hostility, shall I say, to the way you were doing things, and perhaps a lot of misinformation about patenting genomes.
Yeah.
And one of the things I get out of your book is that you're quite clear that you never went into it to patent genomes and make money. How did that myth arise then?
Yeah. Well, just think if you were head of a multi-billion-dollar government or Wellcome Trust program, and somebody came along and said, I can do your program in 9 months for a tiny fraction of what you're spending, you'd probably be pretty annoyed too. It happens throughout history when people get phase shifted by new thinking and new technology and they're unwilling to change. So it's unfortunately, I think it's human nature. I'm sure it happened when Henry Ford developed mass production automobiles and replaced horse and buggies. The buggy manufacturers probably weren't too happy with him.
But I noticed that when you went over towards private funding, that came with its own sort of set of problems as well, didn't it? It wasn't like somebody just said, as you, I think, had hoped, correct me if I'm wrong, Craig, here is the money, just please go get on with it. How was that to work in?
Yeah, there's definitely no free lunch in the world. And, everybody wants something for their money. The foundations and governments want credit. The businessmen want credit and control. So there's trade-offs with every approach, but I think it is pretty extraordinary that private investment money went into the biggest public project ever, made the human sequence available to the world for free. I mean, that's pretty extraordinary. In fact, it turns out private industry does things like this not infrequently. Academics like to think they're the only ones that do anything in that vein. And quite often they block money that goes to innovation. So it's fortunately there are occasionally alternatives for some of us to follow.
Do you think that is your favored approach for the future of science funding?
Well, all the breakthroughs that my teams and I have made have been because we had independent money to pursue it. It didn't matter whether it was ESTs, the first genomes in history that we did, the human genome, what we've done with the environment, and now what we're doing with synthetic genomics. All of those started with some independent resources to prove the idea worked. Once you prove an idea works, it's easier to get government funding, ironically.
Right. Yeah.
So I think I've been fortunate in finding ways to get money to do those pilot experiments to prove that the ideas work. I like to convince people by having the experimental data, not by making arguments.
So you left Celera after the Human Genome Project was completed, and you went to the ocean, which is your other passion, I think, on your yacht Sorcerer II. And it's like a step up in scale, isn't it?
Yeah.
Here is the human genome, now we're going to sequence the planet. I mean, what was your thinking behind that?
Well, we knew that the techniques that we had, the mathematics was able of solving multiple genomic equations simultaneously. At the same time, trying to look around after doing the human genome, myself and my team with Ham Smith and others, obviously we established a certain amount of scientific credibility, and we had the choice to work on whatever we wanted to work on. And I felt and we felt that what we're doing to our environment is the most pressing issue facing humanity. We can't keep doing what we're doing, so we thought maybe our tools would enable us to have a better understanding of the environment and even lead to new discoveries how to protect it better.
How do you see the discoveries that you are making with synthetic genomics actually mapping onto solving environmental problems?
Well, the biggest problem we have is the CO2 building up in the atmosphere, primarily from burning oil and coal. So we're taking ancient carbon out of the ground, we're burning it, and it ends up in our atmosphere. I mean, here we're sitting in this beautiful setting If things continue the way they go, this will be underwater in the future. With climate change, it's just not a matter of places getting warmer. We're already having food shortages in the world. So this is not an option. I mean, humanity can keep going down this track and slowly destroy itself, or we can use the tools of modern science to try and change the problem. And so we're trying to use biology to come up with new sources of fuel so we can have replacements for oil and coal on a renewable basis.
So is that like moving to a hydrogen economy or something different?
I think it's unlikely we'll get to a hydrogen economy, but I think we'll be in a CO2 economy. We can take the carbon from carbon dioxide and convert it into fuels. I mean, that's what plants do with photosynthesis. Yeah. They convert the CO2 into sugar, which you can then burn, but we're trying to make it much more efficient, going right from CO2 into fuel production. If we can do that, then we have a renewable source.
You are obviously most well known for your work in the Human Genome Project, but something that you did recently has hit the headlines, which is the development of a synthetic chromosome from raw laboratory chemical. Do you see that as part and parcel of your vision for applying genomic science to environmental problems, the ability to create this kind of life?
It's the tool set that will be used by tens of thousands of scientists to solve all kinds of problems, not just the fuel problem, but the entire petrochemical industry is based on taking oil as the raw material, making clothing, carpet, plastics. There's the potential to replace that entire industry, starting with sugar, starting with carbon dioxide, using biology to drive these processes. The ability to now write the genetic code allows us to harness this power in a very thoughtful fashion to change metabolisms of cells to do these processes in very efficient ways. So I think it's essential for the future that this new technology is being developed.
What would you say to the lobby who say that interfering with the natural order of things, which is crystallized in the substance of DNA, is somehow the wrong thing to do? What would your response to that be?
It's not sufficient just to have scientists making the breakthroughs if there's not sufficient education in the public to understand what the science means, what the alternatives are, what are the choices being made. The best science could be eliminated by mindless fear.
There is currently in the United Kingdom anyhow a debate, particularly amongst secondary school teachers, about how science should be taught. Should we teach science as individual subjects, physics, biology, chemistry, get to know the basics, build up from there? Or there is a movement afoot to try and enthuse students by telling them what science can do. How would you view that debate? What would you say is the most important of those two things to do?
Well, I think there's other alternatives as well. I think it needs to be taught in a much more progressive fashion, teaching kids that they can solve problems. We have a genomic bus. It's a mobile laboratory out of my institute in Maryland that teaches secondary kids, pre-high school kids, introduces them to solving problems. They do CSI type crime scenes that they solve by using DNA evidence. Okay. And these kids just get really into it. They love it. It's an introduction to science in a way that's not memorizing anything, but it's actually solving problems, and I think it can be very effective.
And so they actually see the application of science immediately up front in a way that they're familiar with from the telly.
That's right.
And then presumably they can be inspired to go back and put in the kind of basic building blocks afterwards to enable them to get up there and do that. Is that the way you see that?
I mean, my own career shows that you can go back and learn the fundamental information later. I essentially started my education over from scratch after I got out of Vietnam.
Yeah.
So when you're motivated to learn and you need to know the facts, you can get them. Also, we all have the advantage of, getting anything off the internet when
Yeah.
you want to know the facts.
Yeah.
Sorting out what the facts mean, the evidence-based decision making, those are the things that if we learn in school, it's easy to find the facts.
It seems to me then that what you're really saying is what the central thing of what should be taught is the scientific method, which goes all the way back to Bacon.
Well, I even have a different view of the scientific method. I think the way it's taught is not how science is really done.
No.
This notion that you have an hypothesis and you go out and test it. Most science is largely empirical. We don't know enough about our universe to ask intelligent questions about it. I mean, that's what we did with the ocean expedition. We just went out to see with the new tools what we could see. And the Challenger took samples from the bottom of the ocean every 200 miles around the globe because the idea was there was no life below a certain depth, and so it was testing that.
What is the single most exciting discovery that's come out of the Sorcerer II expedition for you, do you think?
Well, it's hard to get down to a single one, but if you had to summarize it, the extent of diversity, the amount of life on this planet. In one paper last year, we more than doubled all the genes known to science in a single paper.
That is...
We added 6 million new genes, and that's just from the first quarter of the expedition.
Yeah.
So that's pretty extraordinary, but finding out that all these organisms live by capturing energy from sunlight, not in a photosynthesis pathway, but a totally different mechanism, and that's why there's so much diversity there. Tells us new ways of capturing CO2 back, how life in the ocean works. So, you know, we look out at the ocean, we haven't known fundamentally how it's been working until we've had some of this data. So these are just asking simple questions. Any school child can go take a glass of seawater and make more discoveries in that than in previous decades.
One of the things that you said in your Richard Dimbleby lecture last year was that science makes some people shut down their brains. How would you go about getting them to restart their brains about science if it was left up to you?
With my lectures, I don't give essentially a different lecture to the lay public than I do to sophisticated scientists. And if you challenge people to think, I have high expectations for the public. They can think and understand this. And people that have never had any science before, listening to a sophisticated genomics lecture, they say they can understand 80 or 90% of it. So I think a lot of science talks down to the public. It's important not to do that. And if you teach it as problem solving, we're trying to solve the problems of the planet. That's something I think most people can relate to, and in my experience get pretty excited and fascinated by it.
You've come up through the scientific hierarchy from unconventional beginnings and probably by an unconventional route, which I think suggests that your approach to science is highly individualistic. Do you think that other people could follow that route if they wanted to?
Absolutely. I think it's, there's a lot brighter people than I am in the field of science. It's more of a question of asking questions the right way and being willing to take risk. If you're risk adverse, you shouldn't be in science.
Right.
Because the whole point of science is to understand the unknown. But most governments don't fund scientists to study the unknown. You have to do that on your own somehow and then come back into what is fundable. So I think the entire science establishment can change by rewarding people who work in multidisciplinary teams, by rewarding people if they make major breakthroughs that change society, not just somewhat esoteric findings that may or may not be of any benefit. There's a continuum. People used to separate academic science from applied science. Now we're a continuum, and we have to use all the tools at hand to solve these problems of society going forward. Providing food, drinkable water, shelter, and fuel for soon to be 9 billion people is the largest challenge humanity's ever faced. But I think science needs to change to be integrated. So maybe having a physics department and a chemistry department and a biology department is not the right approach, because we combine all those in the approaches that we're taking and the teams that we're building. The physicists have to learn how to talk to the biologist and to the chemist. And if they all work together and you throw in a little bit of computer algorithm development and building new computers, you have teams that can tackle most problems.
Okay, so this is your kind of continuum idea. The old notion of biology, physics, chemistry, they're all now blurring together, and I mean, you're the arch exponent of that, I think.
Stovepipes, as they have been in academia, the academic institutions that keep the stovepipes will largely become irrelevant going forward.
Do you think that science fiction is relevant to inspiring young people to become interested in science?
Depends on the science fiction. It can be. I think there's too much fantasy world today versus helping all of us understand the real world. And to some extent, nobody's surprised about any development in science because they're so driven constantly with movies and television from the fantasy world. At the same time, great science fiction can be a wonderful extension of the imagination and have people think of what the future might be. It can be great literature or it can be junk literature, just like any other area.
Did you read science fiction as a younger man, or do you still read science fiction? Does it influence you in any way?
I used to read science fiction. Right now I read science fiction and reviewing some of my colleagues' papers.
Right. One of the books that you said that you did read as a younger man was Jim Watson's book, The Double Helix.
Yeah?
And one of the parallels which struck me from reading your autobiography is that genetic research seems to stir up more controversy, pound for pound, than most other sciences. I just wondered if you had a view on why that might be.
Well, I haven't met a person that doesn't have a genome that doesn't exist due to genetics, so it's something everybody can relate to. It is part of our lives. I can't imagine people not asking the question, about traits from their parents. Where did I get this trait? We are a genetic species. We live in a genetic world, so there's probably no science that relates back to us more than genetics. I think that's why there was so much excitement about getting the human genome decoded, and maybe people related to it also because there was a competition. That's something innately human that humans can understand, and even if they didn't understand the science, they understood that it was important and exciting. And with all the new information coming available, the lay person is going to have more information about their own lives than they might have ever dreamed about going forward in the future. So these are very exciting times, and as we go from limited sets of information and limited understanding, you can have controversial ideas. My goal has always been to move to scientific fact, so there'd be real, genuine understanding and elimination of controversy.
Do you think there'll ever be a day when we can actually create complex organisms from scratch? I mean, if you've started with a synthetic chromosome, is there any limit to the complexity of the organism that you might be able to create?
I mean, multicellular species like plants, I think, will be part of the future of design because it's so critical to produce food and energy sources that plants will play a key role in. How long it takes to get to, we're still trying to understand various plant genomes that works following well behind what's happening at the forefront of microorganisms, but I think eventually we'll get there, and it's a critical step to take. As I said, we have a food shortage now in many countries. As the population grows, we need a way to produce a lot more food than the world can produce today.
People will ask Craig Venter, what's next?
I think what's next is trying to put these ideas into action. It's an idea right now that this is the century of biology. We need to prove it's the century of biology by having biology really contribute to these solutions, not just the discussion of them. So that's the challenge facing all of us right now, is coming up with fuels based on biology that really make a difference, coming up with new medicines, coming up with new sources of food. If that happens, I think the world will be a different place.
Do you think that there will be a time when we have tailored medicines for specific diseases and perhaps even for specific individuals?
No. What we will have is, if you understand your genetic code better, you will understand from the medicines that are available which ones you should take, which ones you shouldn't take, which ones are good for you, which ones are bad for you. So it will be individualized medicine by you making choices based on the knowledge of your own physiology, what you should do, versus something being designed for you. I think that's very unlikely.
I mean, you're one of the very few individuals on this planet whose genome, whose diploid genome has been sequenced. Does that bother you, knowing so much about yourself?
Well, it bothers me that I don't know more because we can't interpret it. So there's now 2 genomes done, myself and Jim Watson. His was just published a few weeks ago. The goal is to get it to tens of thousands and then hundreds of thousands and millions.
Okay.
So only at that stage will we be really able to interpret it. There's a lot of data out there. What we need to turn that into understanding.
Craig Venter, thank you very much indeed.
Nice talking to you.